Okay, thanks, Michael. Yeah, I'm Dave Crooks. I work in the Aquatic Ecology and Management Research Group, and I'll just run through some case studies today. So, yeah, very much snapshots. This first slide is just some work I've been doing as part of the NERP, Northern Australia Hub. And that yellow dot you can see moving around is the movement of one of our radio-tagged barramundi during the wet season. So we tagged these fish in October 2013 and watched where they went throughout the wet season. And this was done over at Kakadu in the South Alligator floodplain around Yellow Waters area. And we used the helicopter to track the fish's movements. So out of this work, we found out a lot about how fish use floodplains, and basically the message was they do use them and they use them a lot. One really surprising aspect of this the information we got was that the fish, although they move really extensively over the wet season, at the end, as the waters recede, they were going home back to the exact same billabong where we originally tagged them. So really interesting new information on barramundi and catfish. And one of that bottom point, with barramundi, we found very little exchange between the upper freshwater reaches and the estuary. And that's really interesting because barramundi are a diadromous fish and they have to have salinities greater than 20 parts per thousand before they can breed. And we're finding these fish are moving out all over freshwater but coming back to freshwater. And so we're thinking, well, hang on, some of these fish are really big. They should be going down the estuary to spawn, but they weren't doing it. So another aspect of the research we've been doing under NERP has been to look at the otolith chemistry. So otoliths are the ear stones of the fish. They use for hearing and balance in fish, but they also function as like a chronometer. So it's similar to tree rings. This is the otolith. Can I use it? Yeah. This is an otolith here. That's been sectioned, and there's the annual growth increments, those lines, and that's the core. So we can run a laser transect from the core to the edge and work out the whole salinity history of that fish over its life. So we've done this for a whole range of rivers across the NT and found that we have highly variable salinity histories for individual fish within systems. Many of barramundi that you catch will have never been to freshwater at all, so they aren't actually diadromous and they're not following the standard model. And then many of the freshwater residents, they can be really big fish, but they can have been mature for years and never gone down and spawned during their lives. So this is a big fish that we caught out of yellow water that I'm holding, and I'm 2 metres tall, so that fish is half my size. It's over 1 metre long. We aged that fish as 7 years, and you can see this is the otolith chemistry results. It stayed in salt water down the bottom of the estuary for the first 2 years of life. It then transitioned. Above that line is fresh water. This fish would have matured at around 3 to 4 years of age, but it didn't go down to spawn. And they're hermaphroditic, these guys, so it didn't spawn as a male at all. Because I didn't say, but when we dissected that fish, it was a big mature female. So that fish had been mature for probably 5 years, had changed sex without ever spawning, and is now, we caught it in fresh water, and it's 1 metre long. So it's raised some really interesting questions about barramundi life history, and really, I guess, thrown some, it's got management implications for how you would manage the fisheries, and we might have to rethink the life history model for this very important species. Okay, the next project I'll talk about is the Daly River Fish and Flows project. So this is a project that was originally undertaken under TRAC. It's then supported by NERP, Northern Australia Hub. It's been supported by NT Fisheries and Department of Land and Resource Management. It's been collaboration with researchers from Griffith Uni and very strong involvement from traditional owners in the Daly River area. So this project's aims are to determine the influence of flows on freshwater fish, and it's had a very strong Indigenous knowledge component within the activities. It's been going now for 10 years, so it's one of the most comprehensive long-term data sets in Northern Australia, probably even in Australia now. And it's had a lot of outputs over those 10 years. There's been 24 papers that we know of that have used the data and generated, and 12 of those have had the Daly as really a major part of the analysis. Just some sort of key findings. There's been 98 species of fish recorded in the freshwater reaches of the Daly. So compared to the rest of Australia, that is really, really high number, and they're all native. There is not a single introduced species of fish in the catchment. The work's shown that there's a real high dependency on connectivity. These fish are moving all around the catchment, both across from freshwater into the estuary, but also between the main channel and tributaries. So it's very key to the functioning of the system to maintain connectivity. The seasonality of the flow regime is absolutely critical in influencing fish abundance and community structure. And in particular, the work has found that some species are very vulnerable to possible effects of water resource development. There are species in the Daly who are really reliant, for instance, on riffles, and if they become dewatered, these shallow, fast-flowing zones, due to water obstruction, then that could threaten those species. So it's the information that has been generated has really been feeding into natural resource management, particularly around water. And finally, the Indigenous ecological knowledge has been documented as part of the project. There's been outputs such as seasonal calendars that Michael talked about. And the information has also been very complementary to standard scientific information. For instance, Indigenous ecological knowledge was incorporated into Bayesian belief network models of the response of fish to changes in flow regime. And it's also been used to document things like the economic and social importance of fish for Indigenous communities. So there's been a range of outputs around the Indigenous knowledge as well. My next quick snapshot is some work that Erica Garcia has been leading on tropical river food webs. And this work is aiming to examine the importance of variability in top-down and bottom-up processes in food webs, and then to sort of scale this information up to the ecosystem level to see how these processes play out at large scales. The work's been conducted and supported through TRACK, with support from Department of Land and Resource Management, and it's also a collaboration, a larger collaboration with US scientists as part of this program called SCALA, which is looking at these food web issues and across multiple scales. And it's been going now since 2008. So the summary of this is really the way the project was done. It was an experimental manipulation in streams. Fish were excluded. This was mainly done in Litchfield National Park. And in areas where fish was excluded from the areas, from the stream, they had found higher macroinvertebrate abundance, and benthic algae production was increased. And similarly, by introducing nutrients as a bottom-up control, that stimulated greater benthic algal production. So the conclusion was that there's important implications for health monitoring. And if we have changes to our rivers that increase nutrient loadings or potentially lose different species of fish, this is likely to have significant knock-on effects throughout the food web. Okay, my last task to fly through in 11 minutes is to talk about the Marine Biodiversity Hub. So Peter Kine would have given this talk, but he's in the field at the moment. So I'll try and do it a bit of justice. Peter was working on the NERP Marine Biodiversity Hub, which is just coming to an end now, and it's similar to the NESP Northern Hub. It's kicking off a new round, and it's going through, being directed through University of Tasmania. And as you can see, Charles Darwin Uni is one of quite a big group of players in the marine science area. Pete's input to the NERP and upcoming NESP is really around supporting the management of threatened and rare species. There aren't a lot of threatened and listed marine species in Australia, but we have 3 of them here in the Northern Territory, and they're all riverine sharks, and that's Pete's specialty. And the way he'll be conducting the research is there's a collaborative team that's undertaking this work. CDU's contribution is through Pete Kine and Pierre Futry, who's a NAMRA postdoc and supported by NERP NESP, and Kate Buckley, who's doing work on the conservation of sawfish. And they're working with CSIRO researchers. NT Fisheries has had a very strong input on the logistical side, as well as Kakadu National Park and Mullik Ranges. And there are 4 approaches that they're using in this research. The first is to go out and survey what's there. There's been a dearth of knowledge about the distributions of these species until this recent work. So they're starting to build up really good pictures now of the distributions of these threatened species, and that's feeding into things like EPBC listings, etc. There's a very extensive acoustic telemetry component looking at the movements of these animals, and this has been complemented by population genetics work, which is looking at genetic structure of them over the larger scales and over longer time frames. Probably the most innovative part of this project is the close kin mark recapture, and that's using next-gen sequencing techniques to look at relationships between half-siblings in order to try and come up with an effective population size estimate. So that's really important for understanding just how threatened these species are.